Welcome to our exploration of problem solving in life sciences. In this video, we'll uncover how scientists tackle real-world challenges from understanding diseases to conserving ecosystems. Problem solving is at the heart of life sciences, driving innovation and discovery. Whether you're a student, researcher, or simply curious, mastering these skills can open doors to new opportunities. Let's embark on this journey together, starting with the basics of identifying and defining scientific problems. As we move forward, you'll see how structured thinking and creativity combine to address some of the world's most pressing issues. Every scientific journey begins with a clear definition of the problem. In life sciences, this could mean identifying the cause of a disease outbreak, understanding why a species is declining, or figuring out how to improve crop yields. Defining the problem precisely helps researchers focus their efforts and resources efficiently. It's not just about stating what's wrong, but also understanding the context and implication. By asking the right questions and gathering background information, scientists lay a strong foundation for effective problem solving. This step ensures that the solutions developed are relevant and impactful. Once the problem is defined, the next step is gathering information. In life sciences, this involves reviewing scientific literature, collecting data from experiments, and consulting experts. Accurate and comprehensive information is crucial for understanding the problem's scope and potential solutions. For example, when studying a new virus, researchers analyze genetic sequences, patient data, and previous outbreaks. This process helps build a complete picture, ensuring that solutions are based on solid evidence. Gathering information also highlights gaps in knowledge, guiding further research and experimentation. With information in hand, scientists move on to formulating hypotheses. A hypothesis is an educated guess that explains the problem or predicts an outcome. In life sciences, hypotheses guide experiments and observations. For instance, a researcher might hypothesize that a certain bacteria causes a disease. This step is all about creativity and logical reasoning, as scientists propose explanations that can be tested. Well-crafted hypotheses are specific, testable, and based on existing knowledge, setting the stage for meaningful investigation. Designing experiments is where ideas are put to the test. In life sciences, this means creating controlled studies to examine hypotheses. Scientists carefully plan variables, controls, and methods to ensure reliable results. For example, testing a new drug involves comparing treated and untreated groups under similar conditions. Good experimental design minimizes bias and errors, making findings more trustworthy. This stage requires attention to detail and a solid understanding of scientific principles. The results from these experiments will provide the evidence needed to support or refute the hypothesis. After designing experiments, the next step is collecting data. In life sciences, data can come from laboratory tests, field observations, or surveys. Accurate data collection is essential for drawing valid conclusions. Scientists use various tools and technologies, from microscopes to DNA sequencers, to gather precise measurements. Consistency and accuracy are key, as even small errors can affect results. By following standardized protocols, researchers ensure that their data is reliable and can be compared across studies. Once data is collected, it's time to analyze the results. In life sciences, this often involves statistical methods to determine if findings are significant. Scientists look for patterns, relationships, and anomalies in the data. For example, they might compare infection rates between groups or track changes in gene expression. Proper analysis helps researchers understand what the data reveals about the original problem. This step is crucial for drawing meaningful conclusions and guiding future actions. After analyzing the results, scientists draw conclusions about their hypotheses. In life sciences, conclusions must be supported by evidence and consider alternative explanations. For example, if a drug reduces symptoms in a test group, researchers evaluate whether the effect is real or due to chance. Drawing conclusions is not the end of the process. It often leads to new questions and further investigation. This reflective approach helps science advance, building on each discovery. Effective communication is vital in life sciences. Scientists share their findings through reports, presentations, and publications, allowing others to learn and build upon their work. Clear communication ensures that discoveries benefit society, from new medical treatments to environmental policies. Researchers use visuals, summaries, and detailed explanations to make their results accessible. Collaboration and peer review further strengthen the impact of scientific problem solving. Ultimate goal of problem solving in life sciences is to apply solutions in real-world contexts. This might mean developing new medicines, improving agricultural practices, or protecting endangered species. Implementation requires teamwork, resources, and ongoing evaluation to ensure effectiveness. Scientists often work with policymakers, industry, and communities to turn discoveries into practical benefits. Real-world application brings science to life, making a tangible difference in health, environment, and society. Problem solving in life sciences is not without obstacles. Researchers face challenges such as limited resources, complex systems, and unexpected results. Overcoming these hurdles requires resilience, adaptability, and creative thinking. Scientists often revise their approaches, seek new collaborations, or develop innovative technologies to move forward. Each challenge is an opportunity to learn and improve, strengthening the scientific process. Let's look at a real-world example: solving a disease outbreak. Scientists begin by identifying the pathogen, tracing its spread, and collecting patient data. They formulate hypotheses about transmission and test interventions, such as vaccines or public health measures. By analyzing results and communicating findings, they help control the outbreak and prevent future cases. This case study highlights the step-by-step approach and teamwork essential in life sciences problem solving. Another example is conservation biology. When a species is at risk, scientists define the problem by studying population trends and threats. They gather data from fieldwork, formulate hypotheses about causes, and design interventions like habitat restoration. By monitoring results and adapting strategies, conservationists work to protect biodiversity. This process demonstrates how structured problem solving leads to effective action in life sciences. Successful problem solving in life sciences requires a blend of skills. Critical thinking, creativity, collaboration, and communication are all essential. Scientists must stay curious, open-minded, and persistent, constantly learning from new information and experiences. Developing these skills not only helps solve scientific problems, but also prepares individuals for a wide range of careers and challenges. Summary: problem solving in life sciences is a dynamic and rewarding process. By defining problems, gathering information, testing ideas, and applying solutions, scientists make a real difference in the world. Whether addressing health crises or protecting the environment, these skills are more important than ever. We hope this overview inspires you to approach challenges with curiosity and determination, knowing that every problem is an opportunity for discovery.